Hoy, en Toquin TEC, Valeria Bosio se encuentra con Venki Ramakrishnan, Premio Nobel de Química 2009 y actual presidente de la Royal Society, para repasar su vida y su trabajo épico sobre la estructura y funcionamiento de los ribosomas.
Professor Venki Ramakrishnan, very welcome to Argentina.
Muchas gracias.
You surprised me. Okay, so along with another researchers from the United States, Professor Thomas States, and from Israel, Professor Ada Yonath, you were awarded in 2009 with the Nobel Prize in Chemistry. And the title of the Nobel Prize says, for discovering about how DNA code is translated to life. I hear that, and I hear this, and my skin bristles. You know what I mean? Really, it's huge. So, could you tell me how did you do this discovery? How it's about?
So everybody thinks they know what DNA means, and they think they know what genes mean. But actually, if you ask the ordinary non-molecular biologist what exactly is DNA or what exactly is a gene, they won't be able to tell you. They just know, oh, it's this thing that we inherit from our parents. You can have good genes or bad genes, and you can get mutations and you can get genetic disease, but they don't know what it actually means. And what it is our genes are sections of DNA that contain information on how to make a particular protein. And proteins are long chains of amino acids. So amino acids are little chemical units, and they're joined together to make a protein. And when the protein chain is made, it folds up into a particular three-dimensional shape, which is itself a miracle. It is as if you write on a strip of paper a sentence, and depending on the sentence, the strip folds up into a particular shape. And if you write a different sentence, it folds into a different shape. It's that amazing. But that is how life works. But DNA itself only contains the information. So it's like having a recipe or a blueprint. If you don't have a cook, a recipe is no use unless somebody actually uses the recipe and can make the cake or whatever the dish is. And same thing with a blueprint. Unless you have a factory to use the blueprint to make the product, the blueprint by itself is essentially silent. And in all life forms, the information in our genes is translated to make a protein by a large molecular assembly called the ribosome. It is, in ordinary terms, it's tiny. If you take a single strand of your hair and across the width of your hair, you can line up 20,000 ribosomes. So it's very, very small, okay? But in molecular terms, it's enormous because it has 1 million atoms. Each ribosome has 1 million or more atoms. So the discovery of the ribosome took place in around 1950s, but it took a very long time to understand exactly what it looked like in atomic terms. And that was this long process that resulted in the atomic structure that then resulted in the Nobel Prize.
So, as you said, it's very exciting how this kind of very complex structure in 3D is working all together with a specific function to do something important for the cell, the most important in the cell. And it's hard to imagine as well, how did you get in that field?
Yeah, so it was a long journey, and I actually describe it in a book called Gene Machine that I've recently written about the whole race for the ribosome structure. And when I graduated in India, I was a physics undergraduate, and I was 19 years old when I graduated, and I decided to go and get a PhD in physics from the United States. And I went to Ohio University, but in the middle of my graduate course, in the middle of my PhD, I had already lost interest in what I was doing.
Well, I think that this is very common, right? Between the PhD students,
it can
it's
be
something
common.
that,
Yeah, I mean, It's not uncommon, that's true.
Yeah.
But often what happens is people leave science. They go off and do other things. But in the US, it was possible to be flexible. And what I did was, when I got my PhD in physics, I finished my PhD. But as soon as I finished, I went to graduate school again. And this time I went to graduate school in biology at the University of California in San Diego. And, but I didn't know any biology. So when I went to graduate school, I even took, in my first year, I took a series of undergraduate courses to, you know,
to start again.
To start again. It was almost like starting over. And then after 2 years of graduate studies in biology, I realized I had enough background in biology that I didn't need a second PhD. So I saw an article in Scientific American about the ribosome and using a physical technique to solve to study a ribosomal problem. And since I had a physics background,
you were prepared.
And I also knew that the ribosome was quite important because I'd learned enough biology. So I wrote to them, and they agreed to offer me a postdoc. And so I went to Yale University to work on the ribosome in Peter Moore's lab, and that's how I got into the ribosome. And
sorry, your father was a researcher as well.
Yes, he was a researcher. Yes.
Yes,
and my mother as well.
And your mother, yes. They were collaborating together.
They did collaborate for a large part of their lives. So when my parents married, actually, when I was born, my father was in the United States, and I didn't see him until I was 6 months old. You know, when he came back from his postdoc. And then he and my mother again left when I was about a year old, and then didn't come back until I was about 2.5 or something. So they were quite a scientific couple. And then my father felt my mother was maybe wasting her talent, so he encouraged her to go to get a PhD. So she left me and my father when I was about 3 or 3.5 to go to McGill University in Canada to get a PhD. And she finished her PhD very quickly, in about 18 months, which is quite fast.
A record, yes.
And then came back. And then after she came back, even though her degree was in psychology, she and my father eventually started working on nutrition and you know, its malnutrition and its effects on mental health.
So it's not related, the subject, the field with you, but
no, and
but yes, the ambience.
In fact, part of the reason, I think I didn't want to have anything to do with biology. I thought it was quite boring.
This is because my question, right? Because you started with physics, but
I thought it was quite
how
boring.
did you realize that biology was?
Well, there were several things. One is, you know, when I was taught biology as a school student, and even in my first year of university, I took some biology. It was all about memorizing lots of facts, and you didn't get a sense that it was an intellectual field where there was a problem that you could, you know, attack in an intellectual way. It seemed very empirical compared to physics, which was very beautiful and logical. So I wasn't particularly interested. And one of the things that did interest me was, you know, when I was in the second or third year of undergraduate work, the Double Helix was published. And, you know, the Double Helix is quite a controversial book, but one thing that everybody agrees, even the ones who dislike the book, agrees that it conveys the excitement of going after a big problem and showing the human drama of science, you know, and that made me realize that actually biology is also quite exciting.
Yes. And the title of your Nobel Prize is very exciting. I mean, yes. So going back to the prize, I can guess that you didn't expect it, that prize at your age 10 years ago by now, and for sure you right now remember how was the news for you.
Well, it was a surprise, but not for the reasons you think, okay? Because by the time something gets awarded a Nobel Prize, the people in the field are aware that, you know, it is important enough to get the Nobel Prize. And, you know, many of us were invited to talks all over the world, including many talks to Sweden. Suddenly, you know, after the structures were solved, we started getting invitations to go to Sweden. You know, there's a clear sign that they're taking a look at us, okay, and they want to sort of judge us. And some of these meetings are almost like auditions. They have the character of an audition. They're committee members in the audience and so on. So you're sort of aware that, you know, it might be a prize. So that's not the reason for why I was surprised. There were two reasons. One is there were other prizes that went for the ribosome, and usually almost always I was not one of the ones included in these other prizes.
They were waiting for you.
And the other situation was that at one of these conferences in Sweden, I had a big argument with a Swedish ribosome scientist. And then I found out a few months later that he was appointed to the Nobel Committee for Chemistry. So I thought for sure I would not be one of the ones included. But it says it's to his credit that even though he disagreed with me, he still, you know, must have voted for me. Otherwise, you know, I wouldn't be here, right? So, so I think it shows you that the Swedes are very. You may argue about whether the Nobel Prize is a good thing or not, and about the rule of 3 people and all of this stuff. But I don't think people question their integrity, you know. I think they take their work quite seriously.
But still that, I want to ask you, how is the relation with your students? I mean, I'm very interested in how is your dynamic working with your students before and after the Nobel Prize?
I think that there's been almost no change before and after. My lab has always been very independent. I don't go and check what the student has done every day. I talk to them once in a while to see how things are going, and then we have occasional
Your students are very
meetings.
young, right? I mean, mostly of them, it's young people.
Well, young is a sort of compared to me, they're all young. They're the age of even compared to my son, they're young now. You know, well, once upon a time, they were my age. You know, my students were only a little bit younger than me. Now they're younger than my kid. So it's well, relative.
but this is very good because you can, you know, keep with young people like a feeling. This is very good.
Yeah, no. One of the advantages of working with young people is mentally you don't grow old. At least psychologically, you know, maybe actually you are older, but psychologically you sort of feel, you know, that you're surrounded by young people, so you pretend that you're also young. You know?
Well, but this is very good. I mean, in the good things of youngs, yeah.
Yes, I think
creativity, freedom,
I think, you know, when people are young, they're also very bold and adventurous, and so it's not an accident. If you look at most major scientific discoveries and ask which was the big breakthrough paper, you know, and then say, okay, this was the big breakthrough paper. How old were the authors? And you often find they were quite young. They don't do it when they're much older, you know, because, you know, then you become more conservative, I think.
I used to say when I was younger that if you don't have the Nobel Prize at 23 years old, you will not get it.
Well, maybe that's not been true even in mathematics.
No, not now, but you know what I mean. I want to do a last question related with, that decision that you did when you were young and changed, your field. Really, I wouldn't say dramatically. I mean, it was like a tool for you after you study a new field, but even that, it was a big decision.
Yes, huge.
Yes, a huge decision. And you started again with with undergraduate students. And this is very, very good because it's like you studied again very well. You know what I mean? So
yeah, I think, you know, if you want to learn a new field, you have to have very sound fundamentals.
yes, yes.
And you have to have a good, solid foundation if you want to be good at it. And otherwise, you will learn just the little area that you're going into. If you change a new field and don't get a broad foundation, you'll only learn around that area. You won't have a broad perspective. And many physicists have gone into biology without actually learning a lot of biology. And my feeling is they're often very narrow in the way that they do biology, and they end up not doing very well for the most part. And the physicists who do well in biology are physicists who take the time to really learn what biology is about.
Yeah. So my question is, or the conclusion, I think that you said already, but you know, the people that is not very comfortable with the field that they are, you know, the field that they are choosing, that it's not very late. I mean, it's never very late.
No, it's never too late. I think, you know, also we're living much longer now. So in fact, our working life is quite a bit longer than it used to be. So that also means that, you know, if you're 30 or even in your 40s, there's still time to change and do a completely new thing.
Yeah, just choosing the thing that really motivated you, because you will do better if you are.
And I can tell you, I was using a completely useless technique to study ribosomes. And at the age of 40, I went on sabbatical to learn crystallography. So the technique that I used to solve the ribosome, I didn't even know when I was 40 years old. Okay.
Yeah, yeah.
And so
it was the base of all.
And 7 years later, I was working on the ribosome. 7 years after I learned that technique for the first time. So I think the lesson is, you know, if you want to learn something new, just go ahead and do it. You know, is my advice.
So, how did your investigations go on after the Nobel Prize based on your own discovery?
So, the Nobel Prize can be given at many different stages in your career.
Yes.
Very often they're given long after you've passed your prime. And then these people who win Nobel Prizes, they just go around the world giving lectures and, you know, interviews and so on.
This happened to you?
And, but if you're in the middle and still very active, then you have to be very disciplined. And so my rule was that I wouldn't travel a huge amount. And if you look at the productivity of my lab after the Nobel Prize, in the 5 years after the Nobel Prize, I was more productive than the 5 years before in terms of papers and
Congrats.
so on. And I think, so it's a matter of self-discipline. And people think of the Nobel Prize as an endpoint in somebody's career or science. But it's not an endpoint, it's simply a landmark, it's a recognition of something. But it doesn't mean that the science simply stops there, because science never stops. If you solve one problem, it often raises, you know, 10 different questions, and then you have to go and pursue the next thing. So in the case of the ribosome, the first problem was knowing what the ribosome looks like, and that was the atomic structure, and that was the big race, and the part of the reason we got the Nobel Prize. But once you know what it looks like, you still want to know, first of all, what does it look like in other organisms? You know what it looks like in bacteria, but human ribosomes are much more complex. We also have ribosomes in organelles in our cells called mitochondria. They have their own ribosomes. And parasites have, you know, somewhat different ribosomes. So now we know the structures of many different ribosomes, but we really want to understand how do ribosomes actually work. So then you have to take snapshots of the ribosomes at different stages of the process. Then you want to understand how ribosomes are regulated. How does a cell know to turn on ribosomes and get translation started? And how does the cell know that things are not quite right and you want to shut down ribosomes, maybe even destroy ribosomes? Another question is, how are ribosomes made? because you have this complicated complex of many different proteins and RNA, and they all have to be made in a coordinated way and assembled properly. So the whole business of how ribosomes are actually made is another big problem. So there are lots and lots of problems, and we're working on how ribosomes are regulated, how ribosomes from mitochondria work, and how ribosomes terminate at the end of a sequence, and how the cell knows how to shut down protein synthesis by recognizing when ribosomes have a problem. So those are some of the things that we're doing.
So today you are the president of the Royal Society, and CRISPR, it's something that is revolutionating, the field of science and technology. Can you tell me about this? How is the Royal Society position in front of this, new knowledge?
Yeah. So the Royal Society has been involved in a summit for genome editing. CRISPR is merely the latest and most convenient and perhaps the most powerful way. But even before CRISPR, there were other methods for editing genomes. So it's a general problem of genome editing. And one of the things that we did was we had a summit in Washington with the National Academy of Sciences and the Chinese Academy of Sciences. And then we had a second summit in Hong Kong about last month, actually. And one of the things that has come out of this is a consensus on how CRISPR can be used. So if it's used for research purposes, it's fine. If you want to use it for treatment of diseases, but not for germline editing, but for, editing somatic cells, then you need to be more careful, but there is a way forward and it should be highly regulated. But then there's a more question of germline editing, because you're now trying to change the genes of offspring of future human beings. And that is a much more complex ethical issue. And our feeling is that we don't know enough, either about the technology or even really about genetics, to say, under what circumstances would you want to use germline editing. And at the very least, things like the safety of the technique and the efficiency of the technique need to be much better established before we go down that route. A second point is we also feel this is not a decision that scientists alone can take. Scientists can tell you how safe is a technique and how powerful is a technique, but it's up to society as a whole and up to governments to decide should the technology be used and for what sort of things. And finally, I should say it's an international problem, because if you have different regulatory rules in different countries, people will simply go to the country with the lowest level of regulation to do whatever it is they want. And so this is an area where we have to have international agreement.
Professor Venki Ramakrishnan, very, very thank you for coming, and obviously very welcome to Argentina. And it's really an honor to take this interview for you. So very, very thank you.
Thank you very much.